Begin by cleaning the electrode surfaces and support with methanol or another suitable solvent. Apply a small bead of conductive carbon paint to the upward-facing platinum source electrode. Place a prepared sample of gas diffusion electrode on the upward-facing source electrode. The conductive carbon paint acts as an adhesive. Rotate the cell head 180 degrees and repeat the steps to affix the second gas diffusion electrode. The porous gas diffusion electrode is made from carbon cloth and has a platinum catalyst coating on the side that contacts the membrane sample. The electrode samples are 5 mm wide by 18 mm long. Place the prepared membrane sample between the electrodes. The ideal sample dimensions are 10 mm by 30 mm, although slightly smaller samples could be used. Prior to loading the membrane sample into the MTS cell head, its thickness was measured using a precision thickness gauge. Recording the sample thickness is necessary for subsequent calculation of the membrane conductivity. The electrodes are brought together by tightening the cell head compression cap, which causes the electrode plate to move toward the other electrode. The dial indicator measures the displacement of an integral precision spring with a known spring constant. In this way, we precisely control the clamping pressure imposed on the test sample. The dial indicator is susceptible to damage if exposed to elevated temperatures. Remove the indicator from the cell head when testing above ambient temperatures. The loaded cell head is now ready to be placed into the MTS 740 instrument for testing. Power on the MTS 740 instrument to retract the test chamber locking pin. Screw the cell head all the way into the chamber. The cell head will come to a hard stop such that the FRA connector will be facing the front of the instrument and align with the locking pin. Connect the frequency response analyzer to the cell head. The insulating cell head cover should be used when testing at elevated temperatures. This safety precaution prevents accidental contact with hot metal surfaces and prevents excess thermal loss. With the MTS 740 powered on, start the application software. Warnings, such as this low humidifier water level warning, will appear automatically as conditions warrant. Create a list of experiments to automatically perform the desired test sequence. Experiments permit control of the test cell temperature, humidifier temperature, conditioning time, wet gas and dry gas mix ratio, and the execution of electrochemical measurement experiments, such as impedance spectroscopy. Here is an example of a typical humidity cycle performed over a 6-hour period, displaying the step down and step up in relative humidity. The representative impedance spectra were obtained with the MTS 740 and a Solartron 1260 frequency response analyzer. The high frequency intercept is indicative of the membrane sample's through thickness resistance and is determined by fitting an equivalent circuit model to the impedance data in our ZView software.